Yes, sir. Masters from Dibrugarh and PhD from BIT Mesra, sir. Sir, we and
Excellent.
Naresh work together, PhD. There,
Okay,
in BIT
okay.
Mesra.
Excellent. So, Rangraji, yes, please
Yes, sir.
start.
Okay, sir. Thank you, sir. So, good morning to all of you. Myself, Dr. Naresh Rangra, working as Associate Professor at ISF College of Pharmacy. Welcome you all to the ISFCP sponsored dialogue series. So, today's guest speaker is Dr. Rohit Singh, who is working as Assistant Professor at Department of Pharmaceutical Chemistry, Institute of Pharmaceutical Sciences, University of Lucknow. So before starting the session, I would like to invite our worthy director sir to have some inauguration remarks.
So, thank you very much, Dr. Rangraji. So, first of all, on behalf of ISF Dialogue Series and ISF family, I welcome today's very dynamic, dedicated speakers and researcher, Dr. Rohit Singh ji, from core of my heart. So, Dr. Rohit ji, just we are going to organize this ISF Dialogue Series. The purpose behind of this series to provide the very specific, purified knowledge to the audience, those who are working in academia industry as well as in the research also. So as you know that ISF College of Pharmacy always doing such types of the activity which are the useful for the academia as well as the research. So our IQAC, this is the coordinator, Dr. Siddharth Manji. He is the taking care of all these things of the dialogue series, and all the scientists they are the connecting with their resources either from India as well as from the abroad also. So I hope that today's talk it will be the very useful to the chemistry peoples as well as those person who are the working on the synthesis as well as the some of the other activities which are related to the chemistry. So thank you very much, and I am thankful to the coordinator of today's session, Dr. Rangraji, for provide this opportunity to interact with all of you. And I hope that this talk it will be the very useful to each and everybody. Again, I welcome you, Dr. Rohit ji, and also thankful for accepting our invitation, and you have given your very precious time to our ISF dialogue series. So over to Dr. Rangraji.
Thank you, sir. Thank you for the remarks. So let me introduce to the today's speaker. Dr. Rohit Singh is an alumni of Dibrugarh University, and he attained his PhD from Birla Institute of Technology, Mishra, Ranchi. He has more than 30 publications, including book chapters and books published. He is GATE qualified in 2009, and he also attained his PhD with fellowship UGC MRP. His research areas are drug synthesis and evaluation of N-substituted azole derivatives as potential anti-HIV and antimicrobial agents, computational drug design and molecular docking studies, characterization of synthesized compounds through spectral analysis like NMRs, mass spectroscopy, IR spectroscopy, and including the activities like anti-HIV and antimicrobial screening of synthesized compounds. So his guest of talk is azoles as antimicrobials. So over to you, sir.
Thank you, Dr. Naresh. First of all, I would like to thank Dr. G.D. Gupta, sir, Director Principal, ISF College, Moga, Professor Dr. Narang, Vice Principal, ISF College, Moga, Dr. Siddharth Mehan, Coordinator of IQAC, ISF College of Pharmacy, and Moderator Dr. Naresh Kumar Rangra, ISF College, Moga. So my today's topic is azoles as antimicrobials. So azoles is a very important compound which having some uh, nitrogen containing compounds with a one heteroatom, two heteroatom, three heteroatom as an antimicrobial. I have published several book and book chapter. One of these book azoles as antimicrobial agent published with my guide Dr. Swastika Kamli, which is also a head of department. from Birla Institute of Technology and have one book chapter also. Azoles has potential antimicrobial agent. My guide also did her PhD on these azoles, and we work together as azole as an antimicrobial agent. So first of the things azole, what is the meaning of azole? I would like to describe. Uh,
Sir, sorry to interrupt you, sir. Your slides are not moving. Please check once.
one- Once a minute.
Okay, okay. Now it's moving, sir.
Okay. Is it visible?
Yes, sir. Now it's visible.
Okay.
You can continue.
So first of all, I would like to describe what is azoles. Uh, it is it is a combination of two words. First one is aza. Aza means nitrogen, and oles is a suffix for five-membered ring. If a five-membered ring containing a nitrogen atom, then it is called as azole. If it contains one nitrogen atom, like in case of pyrrole, pyrrole is also called as azole. If it contains two nitrogen, then it is called as diazole. If it contains three nitrogen atom, then it is called as triazole. If it contains four nitrogen atom in its ring, then it's called as tetrazole. And if it contains all of five nitrogen atom in a ring, then it is called as pentazole. So this is a description on the basis of number of nitrogen atom. So azoles, introduction. Azoles are basically five-membered heterocyclic compound containing one or more different atoms, which I said earlier. See the structure here. If if the five-membered ring containing two nitrogen at 1 and 3 position as shown in slide. So it is 1,3-azole, and 1,3-azole is called as imidazole. Similarly, if there is a change in position from 3 to 2, now it is called 1,2-azole. 1,2-azole is called as pyrazole. Similarly, if there is a 3 nitrogen atom, 1, 2, and 3, then that is called as 1, 2, 3 triazole. And if these three are at different position, means 1, 2, and 4. Fourth position, there is nitrogen. So it is called as triazole, but the position is 1, 2, 4 triazole. Similarly, if the nitrogen is at 1, 2, 3, 4 tetrazole, means there is a 4 nitrogen. Only here is a carbon at fifth position. And in next case, all the carbon gets substituted from nitrogen. So there is a 1, 2, 3, 4, 5. So it is called as 1, 2, 3, 4, 5 pentazole. So there are so many kind of azoles which act as a antimicrobial agent. Some of the five-membered azole ring also consist of different other than a nitrogen like a sulfur, oxygen. See here, this is a thiazole, means nitrogen and sulfur separated by a carbon atom. And if it nitrogen get shifted towards second position, then it is called isothiazole. Similarly, there are numerous compounds. Likewise, in place of sulfur, if there is a oxygen, so thiazole will become a oxazole. Similarly, sulfur gets substituted through oxygen, so it is oxazole. And at nitrogen atom also shifted towards second position, then it is called isoxazole. So likewise, thiazole and isoxazole have a numerous compound. Some of the azoles get attached with a ring like a benzene, then it forms a different kind of azole. Means if imidazole, this is imidazole, five-membered ring is imidazole. If it get attached with a benzene ring, then benzene plus imidazole, it will be called as benzimidazole. Likewise, triazole get attached with a benzene, it will called as benzotriazole. This is thiazole get attached with a benzene, called as benzthiazole. And if it is oxazole get attached with a benzene, it is called benzoxazole. So there are numerous azoles which act as an antimicrobial agent. So some of the antimicrobials, we will start with the antibacterial and antifungal agent. Uh, During the last 30 years, azoles act as an antifungal agent, very potential antifungal agent. I will show you some marketed product. This one. This clotrimazole act as a antiseptic cream, antifungal cream. This will be act as a powder azole, ketoconazole 2% cream, ketoconazole as a tablet, clotrimazole as a lotion, clotrimazole as a dusting powder, miconazole as a cream. Means these all are azoles. Whether it is names of clotrimazole, ketoconazole, clotrimazole, or miconazole. It is a very potential antibacterial as well as antifungal derivative. So compounds like clotrimazole, miconazole, ketoconazole, butoconazole, iconazole, fluconazole, fenticonazole, oxyconazole, sulconazole, there are numerous azoles which act as a antibacterial and antifungal agent. And if you will see the structure of all azoles, whether it is clotrima, micona, ketocona, buto, or all are, there is a only modification at N1 position which will attach with a different aromatic ring. I will show you like this one. This is a structure of clotrimazole. You can see the structure. This is the imidazole where I pointed out this one. So this N1 position get attached with a numerous aromatic group like benzene is there, chlorobenzene is there, benzene is there. Means there is substitution at N1 position. Why N3 not get substituted? Because its valency is satisfied. So only substitution takes place at N1 position. You see all the structure. This is clotrimazole. Next one is miconazole. Miconazole is also substituted at N1 position. There is a numerous aromatic group. Likewise, ketoconazole, this substituted here at N1 position. At butoconazole, substitution is here at imidazole. Iconazole, there is also substitution at N1 position at imidazole. Fluconazole, substitution at N1 position. Penticonazole, substitution at N1 position. Oxyconazole, substitution at N1 position. And sulcoconazole, there is also substitution at N1 position. Means in case of imidazole, in the ring of imidazole, N1 position gets substituted with a different kind of aromatic group and form a different kind of azoles which will act as a antifungal, antibacterial, antiviral, antiprotozoal, antihelminthatic, various kind of antimicrobial activity. If you will see the mechanism of action of antifungal agent, that there is simple mechanism that conversion from lanosterol to ergosterol, there is a enzyme 4 alpha demethylase, which will convert lanosterol to ergosterol. But in case of that, azoles get interrupted with this conversion, and lanosterol will not able to convert into ergosterol, and the synthesis of cell membrane of fungus will inhibit. We will see in a mechanism in this one. See here, this is a membrane of fungus. Here is a azole and this is a lanosterol. Lanosterol get attached with the cytochrome P450 at site, whereas the enzyme 14 alpha demethylase will work. But in case of lanosterol, this azole will combine with the cytochrome P450 and it will inhibit the conversion of lanosterol to ergosterol. And if ergosterol will not form, ultimately the synthesis of the stabilization of fungal cell wall will rupture and ultimately fungus will die. So this will work as a antifungal agent like this one. The simple mechanism is the conversion of lanosterol to ergosterol will inhibit by the interaction of azole with cytochrome P450. Next one, antiviral agent. Various azoles also act as an antiviral as we saw previously as an antifungal, antibacterial. This will also work as an antiviral agent. The first known reported compound as antiviral that is capravirine, that is very important. The structure of capravirine is this, and here is a heterocyclic atom is also a imidazole. So imidazole is a very important azole which will work as a antifungal. As we saw the structure in clotrimazole, there is a structure of imidazole, miconazole, imidazole, all are containing imidazole. So imidazole, pyrazole, triazole, these all are very important compounds which will act as a antiviral, anti-HIV, antifungal, antibacterial, antiprotozoal, and so on. And after the modification in capravirine, several SAR, the structure activity relationship by the modification of this X1, this substitution at this ring, X2 substitution at this ring, and substitution at this point, that is Y. If we'll substitute X1, X2, and XY, various numerous compound will also form. The important component for anti-HIV is the reverse transcriptase. There is an enzyme. The transcription process is like that. The formation of messenger RNA from a DNA template is called transcription, and the reversal of means the formation of viral DNA from messenger RNA is called reverse transcriptase. So there is a formation of viral DNA which will inhibit the compound, antiviral compound, that will act as a reverse transcriptase inhibitors, which means it blocks the reverse transcription process which will conduct by a viral DNA, a virus by virus. So there are two types of reverse transcriptase blockers. First one is nucleoside and the second one is non-nucleoside reverse transcriptase inhibitors. The first one is non-nucleoside reverse transcriptase inhibitor. There is a very basic difference between nucleoside and non-nucleoside. Nucleoside, as you all know, nucleoside is a nitrogenous base. There are two types of nucleosides. The first one is purine base and the second one is pyrimidine base. In case of purine base, there is a adenine and guanine, while in case of pyrimidine base, there is a cytosine, thymine, and uracil. If there is a nucleoside compound, then it will contain a purine or pyrimidine base. If it is a non-nucleoside, then other than nitrogenous base, the compound is there. So there are some important non-nucleoside reverse transcriptase inhibitors compounds are DIBO and HEPT. By the modification of these compounds, several other non-nucleosides like nevirapine, delaverdine, and efavirenz, these three are very important non-nucleoside transcriptase inhibitors which will act as an anti-HIV. Most of the non-nucleoside reverse transcriptase inhibitors are basically the reference we take at these three compounds as a nevirapine, delaverdine, and efavirenz. The structure of these three compounds are this one is nevirapine, this one is the efavirenz, and this one is the delaverdine. And its mechanism of action is also like a reverse based on reverse transcription. Means if the virus enters into the cytoplasm, it will act on a nucleus, while previous one this will act on a cell wall. Means antifungal, antimicrobial act on a cell wall or cell membrane, while this one will act on a nucleus. Means reverse transcription will take place at a nucleus position and it will also block the generation of, means the new reverse transcription will block by these non-nucleoside compound. This one is a nucleoside reverse transcriptase inhibitors. Here you can see a nucleoside nitrogenous base. That one is, this one is a pyrimidine. The structure is gyrovodine. Gyrovodine contains a pyrimidine nitrogenous base. Didenosine, this will also contain a purine. This one is a purine. This one is a pyrimidine. Pyrimidine is nitrogenous base. While in case of didenosine, there is a purine nitrogenous base. Gelcitabine also contains a pyrimidine nitrogenous base. Stavudine also contains a pyrimidine nitrogenous base, and lamivudine will also contain a pyrimidine nitrogenous base. The basic difference between these two structures, nucleoside reverse transcriptase inhibitor and non-nucleoside reverse transcriptase inhibitors, is just only a nitrogenous base. Nucleoside, there is a nitrogenous base. Compound is didovodine, didenosine, gelcitabine, stavudine, and lamivudine. While nucleoside, non-nucleoside is nevirapine, efavirenz, delaverdine, PIBO, HEPT like compounds where nitrogenous base is absent. There is a table difference between nucleoside and non-nucleoside reverse transcriptase inhibitors having a some major side effects. Compound like in case of nucleoside reverse transcriptase inhibitors, didovodine, some side effect like osteoporosis, bone marrow anemia, gastrointestinal disturbances, insomnia, neutropenia. In case of didenosine, there is a side effect like pancreatitis, peripheral neuropathy, gastrointestinal disturbances. In case of gelcitabine, peripheral neuropathy, stomatitis. In case of stavudine, peripheral neuropathy, pancreatitis. In case of lamivudine, there is a side effect like nasal symptoms, lactic acidosis, and liver problem. Similarly, in case of non-nucleoside reverse transcriptase inhibitors, in case of nevirapine, there is a symptoms like rash, that is Stevenson-Johnson syndrome, tingling, burning, prickly sensation. In case of delaverdine, rash, excessive tiredness. In case of efavirenz, like convulsion, false positive test of urine, dizziness, and drowsiness. These are some side effects of nucleoside and non-nucleoside reverse transcriptase inhibitors. Basically, the chemical compounds in the synthesis, there is in synthetic compounds, there is a some reactions, some disturbances. If you will take the medicine, there will be some chances of side effects is there possible. This is a site, this is a protein where the nucleoside or non-nucleoside reverse transcript inhibitors get interacted with the protein and will show their work. This is a DNA polymerase active site, non-nucleoside reverse transcriptase inhibitor binding pocket. The length of these is 10 angstrom, where the our active molecule get bind together and will show the activity. As we saw, as we saw, antibacterial, antifungal, antiviral. Now the last one is antiprotozoal compound. Means there is a several kind of azoles which will also act as a antiprotozoal. This is very primitive type of compounds. The structure is nitroimidazole. Imidazoles and nitroimidazoles are very important compounds. By this, the modification are compound called metronidazole, which is marketed as a metrogel, very potential antiprotozoal compound. Nitroimidazole and anti-trichomonas activity were reported in year 1961. That means very previously. And then in 1966, a modified compound of nitroimidazole, that one is metronidazole, was among these compounds. It is exhibited broad anti-protozoal activity and found wide use in case of trichomonasis orally. Metrogel is a very famous marketed product of metronidazole, and this is a nitroimidazole derivative. Means there is also a imidazole. So imidazole shown various activities like antifungal, antibacterial, antiviral, anti-HIV, and now this is showing antiprotozoal. If you modify the metronidazole, means the SAR of this metronidazole, various compound like tinidazole, nimorazole, penidazole compound were synthesized which having shown better activity than metronidazole. Tinidazole is the most potent towards a protozoa, that is ant amoeba histolytica in vivo, while in case of in vitro, fecal amoebiasis and hepatic amoebiasis in experimental animals. Means whether it is metronidazole or tinidazole having a nitroimidazole molecule which will show a better antiprotozoal activity. These are some marketed products which I showed earlier. This one is a antifungal cream, clotrimazole. This will contain a imidazole ring. This is a azole plus that will also contain a imidazole ring. Ketoconazole 2% cream, BP, British Pharmacopoeia, marketed as a keto pack. This will also contain a imidazole ring. Ketoconazole is also available in the form of tablet that will also contain a imidazole ring. Clotrimazole, whether it is lotion or whether it is in the form of dusting powder, it will also contain a imidazole ring. And miconazole nitrate, that is also contain a imidazole. So these are the important products which is having azoles, especially imidazole, as a marketed product. This is some of my publications. Book, the front page of book chapter and book I shown earlier. Thank you. Anyone having any query regarding azoles? If you have any query, please ask. This is very important compound azole, various type of azoles, whether it is diazole, whether it is triazole or tetrazole. Anyone?
Any student from the audience wants to ask any question, you can ask.
If you will go in the synthesis of azoles, you will surely 100% will get a activity where any of activity, because nitrogen, oxygen and sulfur is a very potent hetero element which shown a very effective antimicrobial activity or antibacterial activity, antifungal activity. You will definitely get results. So this is very and very broad spectrum chances is there.
Thank you for this mind-opening section, sir. So it was a wonderful lecture
Thank you
on
very much,
the
Dr. Ranga
topic that
sir.
is.
I'm very thankful to ISF College for giving me such a platform to express my topic here, and I will definitely very eager if anyone having any doubt.
So on behalf of IQAC, ISFCP sir, and ISF College of Pharmacy, I extend a really hearty vote of thanks to our expert speaker, Dr. Rohit Singh, who spared time from his busiest schedule to grace the event. So today we had an opportunity to hear your thoughts on the topic Azoles as antimicrobials, and this will surely be going to encourage us in our future research events, sir. So thank you.